Hello, my name is Kate Bowers, and I'm an associate computational biologist at the Broad Institute in Boston. My presentation is titled Population Genomics Defines the Global Structure and Species Boundary of Cryptococcus deneoformans. Cryptococcosis is a serious fungal infection with significant mortality risk to HIV-positive individuals. It's number one on the World Health Organization's fungal pathogen priority list in the critical priority group. The global burden of HIV-related cryptococcal meningitis is highest in sub-Saharan Africa, and that is the major global location. Cryptococcus is environmentally acquired. People would breathe it in from environmental sources like soil, trees, and avian excreta, where it goes into the lungs and either is fought off by macrophages or, in the case of immunocompromised patients, may develop into cryptococcal meningitis, which can be life-threatening. There are two serotypes of Cryptococcus that are most often responsible for infection. There's Cryptococcus neoformans, which causes 51 to 70% of infections, Cryptococcus deneoformans, which causes 19 to 30% of infections, and then there's hybrid isolates between the two, which are less common. While neoformans is well studied, deneoformans and the hybrids are less well studied, which motivates this project. So first, for our population genomics analysis, we developed a cloud-based pipeline written in workflow development language, or WDL, available on Terra.bio to process raw sequence data for population genomics analysis. We start with sequenced clinical samples, convert them to unmapped BAMs, and then in this Terra pipeline, we pre-process the reads and align them to a reference genome, call variants in each sample BAM, combine calls across all files into just one VCF, do joint genotyping of that, filter the variants, and do quality control checks to make a final VCF that we can use for downstream analysis like GWAS, aneuploidy analysis, copy number variation, and phylogenomics. This pipeline is available on GitHub if you're interested. We like using Terra and this WDL language because it allows us to use automated data organization, linking inputs, outputs, and metadata, call caching, and easy relaunch of failed runs, version control with snapshots, and highly parallel computing. With the system, we sequenced and analyzed a diverse global population of C. deneoformans isolates to characterize the species population structure, genetic diversity, in comparison to Cryptococcus neoformans. These samples ranged from 15 countries, clinical, environmental, and veterinary sources, and over 5 continents. Unlike C. neoformans, however, we see that the deneoformans population does not appear to have any subdivision into lineages or substructures, and no relationships related to isolate type or country of origin, mating type, or hybrids. Deneoformans also shows lower genome-wide diversity than neoformans. Next, we identify a new region of introgression of C. neoformans sequence into C. deneoformans isolates. We find 3 isolates with this new introgression region. You can see here, we align these isolate sequences to a combined reference sequence of neoformans and deneoformans. And then for each isolate, we plot the normalized depth of coverage against each reference genome. You can see here that while this isolate AD682 primarily maps to deneoformans, there's a section where there's a dip in deneoformans coverage and a peak in neoformans coverage, which is this introgression region. So previous studies have found this identity island, which is a 40 kilobase 14 gene introgression of neoformans chromosome 5 into the non-homologous deneoformans chromosome 5. This region is present in all deneoformans isolates. Our newly identified region here is a 144 kilobase introgression, and it actually introduces another copy of the identity island into the homologous deneoformans chromosome 4. It replaces deneoformans genes in that location with their neoformans orthologs. And the linkage between the neoformans chromosome 5 and deneoformans chromosome 4 in these introgress isolates has been confirmed with de novo sequence assembly. Can we learn more about these species by studying differential selection between them? Yes, so we looked at differential selection with the McDonald-Kreitman test, the MKT, and looking at over 6,000 ortholog pairs, we found 18 genes which have significant positive selection. These were related to capsule production, which is an essential virulence factor for Cryptococcus, as well as cell viability, survival inside macrophages, which is essential for infection, antifungal drug resistance, and infection burden. So it's interesting to see what's different between these two serotypes. Finally, deneoformans chromosomes appear to share patterns of aneuploidy with their homologous neoformans chromosomes. So aneuploidy in fungal pathogens can be a route to drug resistance, hence why we are interested. And we find similar rates. 9 to 12% of neoformans clinical isolates are shown to have partial or full aneuploidy, and about 15% of our deneoformans clinical and veterinary isolates showed partial or full aneuploidy. And they appear in the same homologous chromosomes generally. Overall, our sequencing, variant calling, and population genomics analysis of diverse seed deneoformans isolates expand our understanding of global Cryptococcus speciation and genome diversification. Thank you so much for listening to my presentation. Thank you very much.